Yeah, that's very. You're like me with the ocean. That's because that's very, like, I was just kind of like, you know what? I give up, you know, see, it's like, I think I'm going to go over representing functions in scalar series.
Okay, it's noon, so let's get started. Grades are not posted yet. I see way too many happy faces. I'm just kidding. I don't think it's posted yet. Okay, so let's do a quick recap of what we've covered so far. What did we talk about yesterday? At some point, somebody has to explain to me what happens to the brain in 24 hours. Phenotypic sex determination? I like it. Yes. Okay, so what is the key thing that if there was one word that you can associate with phenotypic sex determination that is different from gonadal sex determination, what would that be? Hormones. Hormones, perfect. Hormones, 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 hormones. Okay. So yesterday we finished talking about what's involved with making a testis and making an ovary. We kind of talked about what the different cell types were. We're going to keep following that along in the next coming lectures. Okay. And then we talked about the fact that hormones are being secreted. Okay, and these hormones did a whole bunch of things. We spent time talking about Alfred Joseph's experiments. We talked a little bit towards the end about disorders of sex development, things like that. Okay, so let's see. What hormones are being produced by the fetal gonad? Testosterone is one. Estrogen? Okay. What do we think? Raise your hand if you like testosterone as a fetal hormone being produced. Me too. What about estrogen? Like that one too? Did I talk yesterday about fetal ovaries secreting estrogen? No estrogen. No estrogen whatsoever. Okay. Anti-Müllerian hormone, I like that one. Okay. So yes, we talked about two hormones that had quite a bit of an impact on two ducts. What were the ducts that we talked about? Wolffian and Müllerian. Okay. Name me one structure from the Wolffian duct. The vas deferens. Yep, that's one. Can we name another one? Seminal vesicles, also one. Another one? Epididymis. Is there another one, or are we good? That's it. Just those three. Okay. What about the Müllerian duct? What does the Müllerian duct give rise to? Which one? Uterus. Uterus is one. Oviduct is one. Cervix is one.
And the upper portion of the vagina.
Upper portion of the vagina. Perfect. Very good. Okay. So again, for the exam, know these things. Okay. Just know these structures. Okay. And all these structures we'll talk about, okay? So today we'll talk about male reproductive structures, probably more so on Monday, but and next week we'll definitely talk about the female reproductive structures as well, okay? So all these structures, okay, so then we kind of finished here, okay? So I kind of gave you this, like, it's not really a homework assignment, obviously, but really, if you really want to understand phenotypic or secondary sex determination, you really need to understand how Alfred Joseph's experiments work, okay? Because some of these questions can come up on the exam, okay? And I can be really creative with some of these things. So really understand how this works, okay? So one of the disorders of sex development that we talked about at the end yesterday was this baby that was born that if I hadn't indicated to you whether that it was XX or XY, it was kind of difficult to tell, right? It had a case of what people refer to as ambiguous genitalia, okay? This baby could have been either XX, that was masculinized, or XY, that was undermasculinized. And unless you do a chromosomal test, you will not know which one of these it is. As it turned out, this individual was XX. Okay, do we remember why this XX baby looked somewhat masculinized? Had to do with another organ doing something.
Congenital adrenal hyperplasia?
Yeah, so basically an overactive adrenal gland starting to crank out some testosterone, right? So this testosterone starts floating around, and how is it possible that that causes masculinization of the external genitalia? What happens?
Testosterone.
Yeah, so the testosterone was the first thing that happens with the testosterone.
Gets excreted.
Yeah, it gets excreted by the adrenal gland, and it flows around, and then we had this enzyme.
5-alpha reductase.
Yeah, 5-alpha reductase, and it turns it into supertestosterone, right? DHT. And the reason why that tissue responded to that was because of what? How come that external genitalia responded to DHT? It's because of one thing: the presence of receptors. When we talk about hormones, the only way hormones work if the matching receptors are present and functioning. Make sense? Perfect. Okay. And then the other one is kind of where we left things off. This was kind of that homework assignment to kind of figure out for yourself, do you really understand Alfred Joseph's experiment, right? I showed you a different picture, but initially, this is another individual that has the same condition. So this is a person that has 46 chromosomes. Is that weird? 46 chromosomes? No, that's normal. Okay. But it was 46XY. Is that weird? Well, kind of. For this individual, yes. Okay, that is not what is expected, right? Okay, so this individual is 46XY, okay, and it has something called complete androgen insensitivity syndrome due to a mutation in the androgen receptor. That means the androgen receptor is not working, okay? So let's go through these. How many of you have tried to do this at home before coming to class? Couple? Okay, let's see how far we got with this. So chromosomal sex, that's an easy one, right? Because I pretty much told you. Which one, the chromosomal sex is? XY, perfect. Otherwise, I'm just showing you a naked picture of a woman that's XX, okay? Gonadal sex. So with gonadal sex, I mean, what kind of gonads does this individual have? Does this individual have testes, ovaries, ovotestes, no ovaries, no nothing? 20 testes, you come up with something. Testes, raise your hand if you think it's testes. Raise your hand if you think it's ovaries. Raise your hand if you think there are no gonads that are being developed here. Nobody? Okay. So, why testes? There were quite a few that were very enthusiastic with saying testes. Why?
Because of those two things that when you have the Y chromosome, like force it to do that.
Yeah, so basically we start, this individual is what? XY. So this individual starts with a Y chromosome. That means this individual has what? SRY, okay? What did SRY do? Turn on SOX9. And what did that do? Make a testis, right? It makes a testis. What hormones are involved in making a testis? None. Absolutely not a single hormone is necessary to make a testis. Now, some other species, yes, some fish species, amphibians, you can force hormones and force gonads to change. In humans and mammals, impossible. Making a testis or making a hormone or an ovary is genetic factors, not hormones. I don't care how much testosterone is there. You could kill the individual with testosterone. It's still not making a testis, okay? Okay, so we have SOX9, we make a testis, there we go. Where are the testes in this individual? Can you see them? So where are they? Inside. Okay, so early on in development, just like the ovaries you can find right underneath the kidneys, the testes start there as well. Okay, so one of the things we'll talk about today is the fact that testes actually have to descend and actually have to descend into a scrotum in order to function properly. Okay? So in this case, that never happened, and the testes are still inside. Okay? Okay, so that is the gonadal set. But phenotypic sex, obviously what I mean is not, let's see, this. So XX, testes undescended, not this. I think we all recognize this as a female, right? Even my 10-year-old son, years back when I was putting these slides all together, was kind of curious, wondering why am I staring at this picture of a naked woman? It's the female, okay? The question here is internal genitalia, okay? So, but this is really, okay, apply Alfred Joseph's experiments, okay? What can we find inside? Epididymis, vas deferens, seminal vesicles, uterus, oviduct, okay, cervix, upper portion of the vagina. What is present? Both sets are present. But some of the experiments that Alfred Joost did, both of them are present, right? Okay, so what's present here? Who wants to give this a go? Go for it.
The Mullerian duct.
So the Mullerian duct is present. So basically, when inside, we would have the oviduct, uterus, cervix, upper portion of the vagina. Okay? Who agrees? No worries, we're not going to turn this into a heated discussion or anything like that. You too? Okay. What about the male part? The Wolffian duct? Is there anything from the Wolffian duct? Epididymis, vas deferens, seminal vesicles? Yeah? Okay. So what if I were to tell you that neither is present? Nothing. So let's think about this. This one has testes. The testis secretes what? Testosterone. Let's start with testosterone first. Does the testis secrete testosterone here? Absolutely. What's the problem?
It has no effect.
There's no effect because the receptor is mutated. It doesn't work. Like I said, I don't care how much hormone you have. If you don't have the receptor for it, it's not going to work. It's like any door. You can have all the keys in the world. If there's no lock on that door, it's not going to do anything. Okay? So the Wolffian duct is gone. That's what Alfred Joseph's experiment showed as well. If you add testosterone, it stays. If you remove testosterone, it disappears. Okay? What about the Mullerian duct? Why is that not there? Why is there no uterus, no oviduct, no cervix, and no upper portion of the vagina, meaning the inner portion? This actually, she would have an outer portion of the vagina, really, which is basically a blind ending pouch. Okay? So why wouldn't she have any of that?
Is it because there's no Y and like the SRY stuff prevents that stuff from forming?
Not the SRY specifically, but you're on the right track. Go back again to, we've made a testis, now let's take the next step.
Is it because of the anti-Mullerian forming?
Perfect, perfect. We have a testis. We dealt with testosterone. It doesn't work, but there's still testosterone. The other thing that the fetal testis secrete is anti-Mullerian hormone. Whether the androgen receptor is mutated or not, that has no impact on an receptor for AMH. Different hormones, different receptors. The AMH receptor is perfectly fine. So the fact that the testis secretes AMH, it causes the Mullerian duct to completely disintegrate. So no oviduct, no uterus, no cervix, and no upper portion of the vagina. Make sense? Kind of? Think about this stuff, okay? If it still doesn't make sense after a whole weekend and you're not getting any sleep, then ask me Monday. Yes, questions? Pursue.
She still has the outer portion of the vagina, but no uterus, right? Because there's no Mullerian duct.
Perfect.
Does she have a vaginal canal, I guess?
Yeah, so it's somewhat of a vaginal canal, but it would be closed. A closed pouch, yeah. Yeah.
So if they don't have any plumbing, where do the testes, what are they attached to? Are they just like floating there?
So the testes itself, we'll talk about this in a minute when we talk about testicular descent. So basically, this is a figure that I meant to draw later, but we'll do it right now. Here's the testis. Normally, it's maintained in place by two ligaments. One that I'm not going to even write down because then you'll write it down and you think you need to learn it. It's a cranial suspensory ligament. Leave that alone. The other one is called a gubernaculum that attaches it to the body wall. Okay? The plumbing has nothing to do with keeping it attached. Okay? So there's two different ligaments. Okay? Any other questions about any of this? So start thinking about what would happen if you now have an individual that had an AMH receptor mutation. What would happen, right? Go back, what I'd like you to do is for the exam maybe, go back to Alfred Joseph's experiment, that slide that had these 3 experiments on there. Right next to it, what you think the phenotype externally, what do these bunnies look like, male or female? Okay, we never talked about that. We only talked about the internal stuff. And if you can't figure that out, then ask on Monday. Okay, but try to figure it out yourself first. Okay, so in the other class we had some questions about this. Is this individual fertile? No. Okay. Does the testis produce sperm? No. And even if it did, it could never go anywhere. But when do you think this individual discovered she had an issue?
Doctor's office.
Yeah. When? Very good. In the doctor's office, probably. Why would she go to the doctor in the first place?
No menstruation.
No menstruation, exactly. So how old would she probably have been, right? So this is not at birth. This is not a condition at birth. A baby, when she was born, what would she look like? A baby girl. And a perfectly normal baby girl. Nothing going on. And I told you before, between birth and puberty, there's nothing interesting happening related to reproduction. So probably this individual, by the time you, depends how impatient we are, I guess, but when you're 13, 14, 15, 16, and you still haven't had your first period yet, you're going to go to the doctor and you're going to try to figure out, okay, everybody else in the world seems to be getting their period and I am not, and I should be. Okay? So then we start figuring out the first thing that she found out then was via a genetic test that she in fact is not XX, she is XY. What do you think was the first thing? What are the 2 major things that they would have done for her?
Would they give her estrogen so that she develops secondary sex characteristics?
Perfect. Yeah, like anything else, when girls start puberty, the secretion of estrogen and progesterone and things like that help with the secondary sex characteristic, like specific adipose tissue deposition in the hips and the thighs, breast development, things like that. So they would have started estrogen and hormone treatment. Okay. What's the other thing they probably would have done for her?
Need to leave the testing.
Very good. So the answer to that is no, do you want to remove them? Okay, so the first thing they probably would do is remove the testes. The reason for that is if you leave testes inside the body cavity, there is a very high chance of developing testicular cancer. Okay, so this becomes cancerous tissue. On top of that, because this individual, like even when she would have been 13 or 14 years old, that gonad has been in an environment that would have caused these germ cells just to become abnormal, and she would have been completely infertile. Okay, so it's best just as a preventative measure to take that out. It's not going to be functional. It can become cancerous, so there's really no reason to leave that inside. Okay, cool. Any questions about this stuff? Make sense?
I'm so sorry.
Yeah, no.
But with respect to the secondary sex characteristics and providing a hormone treatment, would she be able to, or he or she or whatever, be able to produce estrogen or progesterone despite not having the hardware to do that?
Yeah, no. So she would have to take these hormone supplements for the rest of her life.
Okay.
Yeah. So really the only source of estrogen and progesterone, there are peripheral tissues that will secrete some estrogen, like adipose tissue, for example. But it is not enough to at least help with the secondary sex characteristics, and progesterone and stuff like that only comes from the ovaries, which she doesn't have. So could she ever become pregnant or carry a baby? Yeah, it's a tricky one because there are some recent reports out there where people have now done uterine transplantation, where you transplant a whole uterus into a woman. And so, yes, science kind of starts spinning out of control here. Normally not, but I guess if you start doing something like that, it is, I guess, theoretically possible. Okay. Any other questions before we move on? Yes.
So does someone who's XX have androgen receptors?
Yes. Androgen receptors is a very good question. Everybody has androgen receptors. And the reason for that is even if women don't have androgen receptors, so they've done that really clearly in mice, for example, many years ago. You mutate the androgen receptor, guess what? Females are subfertile, infertile as well. Okay? There is a role for androgen signaling in females. The only reason why we call testosterone the male hormone and estrogen the female hormone is that normally if you take a blood sample, testosterone levels are higher in men than they are in women. Okay? And estrogen levels are higher in women than they are in men. Both men and women have estrogens, both men and women have testosterone, and in fact, in men, if they cannot make any estrogen, guess what? They're infertile. There's part of the male reproductive tract where estrogen signaling is so important that if it doesn't exist, okay, it causes testicular atrophy. So, in terms of male and female sex hormones, the nomenclature is kind of terrible. It's basically what's floating around in your bloodstream. That's it. Okay? Any other questions? Okay, like I said, go back to Alfred Jones, that slide, try to figure out what the external phenotypes are to really kind of get a handle on this hormones and external differentiation. Okay, let's do male reproductive physiology. We'll kind of go through this. This really is not horrendously complicated. The only complicated part with this is vocabulary, nomenclature, anatomy. Okay, for the rest, this is very simple and straightforward. Okay, what definitely helps is that there's no cycle. That really is a great benefit. Use this kind of as a guide, okay, to kind of see what the main topics are, the main key points. When Dr. Warren talked about the endocrine system, okay, I'm sure that he talked about the hypothalamus, pituitary, and some kind of an organ. For example, the adrenal gland, right? Something coming from the brain, pituitary, and then a signal to the adrenal gland? Or did he not talk about any of that at all? Okay, somebody's nodding yes, so it is just we've forgotten about it, and it sucks because I think for the final exam there's an endocrine section on there too, sorry. Okay, so that is with all these endocrine systems. So you can think of the endocrine system as one thing, but there are different parts to it. Hypothalamus, pituitary, adrenal gland. Hypothalamus, pituitary, thyroid gland. Okay, hypothalamus, pituitary, liver, and growth factors, and bone, and stuff like that. Okay. For reproduction, hypothalamus, pituitary, gonads, testes and ovaries. Okay? So if we start in the brain with the hypothalamus, any hormone released from the hypothalamus, a great rule of thumb is that it ends with RH, releasing hormone. Okay? In this case, the releasing hormone is called GnRH, gonadotropin releasing hormone. Okay? On the exam, the hormones will be abbreviated like that. They're not written out. Okay? So GnRH is gonadotropin releasing hormone. Comes from the hypothalamus. That goes to the anterior pituitary gland. Most of the hormones come from the anterior pituitary gland. There are only two that come from the posterior pituitary gland that we'll talk about next week. And actually one you already talked about. And actually was on the exam, I believe, that you had Wednesday evening.
Is it vasopressin?
There you go. Vasopressin, see? Still there. But that has nothing to do with reproductive physiology. That's kidneys, okay? So the two major hormones from the anterior pituitary gland that I want you to know are LH, or luteinizing hormone, and FSH, follicle stimulating hormone. This name, follicle stimulating hormone, makes sense in females. Makes absolutely no sense in males, because in the testis we cannot find any follicles. There are no such things as follicles in the testis, okay? But it's both in men and women, LH and FSH, okay? These peptide hormones travel through the bloodstream and act on ovaries or testes, depending what you have, okay? LH, generally speaking, acts on the steroid-producing cell. And in the ovary and testis, it acts on the cell that makes testosterone. So LH acting on a specific cell in the ovary and testis causes secretion of testosterone. So, in the testis, where do you think LH acts on? We already kind of talked about this. What was that steroid-producing cell in the testis? Leydig, exactly, Leydig cells, okay? Remember from the lecture yesterday what the female counterpart is of a Leydig cell? Start with a T. Theca cells, theca cells. So theca cells and Leydig cells are this counterpart. LH acts on those cells to cause testosterone secretion. Okay? FSH tends to act on the supporting cells. Yesterday we talked about a population of cells in the gonad that were called somatic support cells. So FSH acts on which cell type in the testis? Remember what we called that one? Named after Enrico. Then starts with an S. Sertoli. Okay, Sertoli cells. What was the female counterpart to Sertoli cells? Remember that one? Starts with a G. Granulosa cells. Granulosa cells. Okay? We'll talk about this stuff. I'm already giving you a lot more information, but that will come back over and over and over again. So don't panic. Okay? And then we get negative feedback. Every endocrine system has negative feedback. It's basically to put a brake on things, otherwise it's going to keep going and going and going. Okay? A terrible slide unless you absolutely love biochemistry. Okay? Just a couple of really quick recaps. One, from the endocrine lectures from Dr. Warren, do we remember where steroid hormones come from? From what chemical? Cholesterol, it's right up there. So is cholesterol good or bad? Well, it's good because we need it. It's a precursor to making steroid hormones, which are absolutely necessary. Okay, it's part of our cell membrane. Okay, so those are good things. Okay, and then cholesterol gets converted, and then the next one that I want you to know in this whole elaborate scheme of enzymes is progesterone. Okay, so one of the first steroid hormones that is being made from cholesterol is progesterone. And progesterone, especially when we talk about female reproductive physiology, progesterone becomes really important. Okay? In men, progesterone really doesn't do much of anything, except it's a precursor hormone to many other things. Okay? So progesterone then gets converted to many, many things, blah, 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 and then eventually we have testosterone. Okay? So in order to make testosterone, you first need cholesterol, turning into progesterone, turning into a bunch of other stuff before you make testosterone. And testosterone itself is a precursor hormone to two things that one of them we already talked about. Okay? So testosterone is actually a precursor to dihydrotestosterone. Right? We had this enzyme called 5-alpha reductase. Okay? 5-alpha reductase. What was DHT again? Provide some context. What are we talking about here? What was important? What did it do? The super testosterone, right? And that was important for what?
Binds to the androgen receptors.
And does what? Yeah, so the male developmental pathway, right? Masculinization of the external genitalia. It causes elongation of the genital tubercle, development of the scrotum, all that stuff. Okay? Testosterone, as this slide shows, in fact is a precursor to estrogen. That's why women have to make testosterone, otherwise they can never make estrogen. Okay? So estrogen, E2 is usually the symbol for estradiol. Okay? The enzyme that's involved with that, aromatase. That's a lot cuter than CYP19A1. Nobody cares for that thing. Okay? So aromatase is an enzyme that will take testosterone and convert it into estradiol. And estradiol released in bloodstream does a whole bunch of things. Okay? Then a couple of other quick things to kind of wrap up this part of the steroid hormones. Androgens is just a collective word for things like testosterone and dihydrotestosterone. Those are androgens. That's why we call the receptor for these things, since it's the same androgen receptor. Estrogens are a collective word for things like estradiol, estrone, estriol. They all bind the same receptor called the estrogen receptor. Okay, so it's just a group name. Okay, and then finally, progesterone is a steroid hormone that obviously that's what we make first in order to make all the other stuff. Okay, and then quickly, steroid hormones. Remember from the endocrine lectures that these are lipophilic, so they like fat. That also means that when it transports through the bloodstream, it needs to be bound to plasma proteins. Okay? It binds receptors that are inside, not on the cell surface, not part of the cell membrane. Okay? So either in the cytoplasm or the nucleus. And usually the effect is that that hormone, that steroid hormone, like testosterone, bound to the androgen receptor, it binds DNA. And it causes transcription. Okay? So these are transcription factors. Okay? Okay, male reproductive structures. Okay, so really basic, some basic anatomy here. We're going to start right here in the testis. So pretend that this classroom is the testis and we are all sperm. And we're going to try to find our way out. That's how we're going to go. Okay? So right here in the testis, the first thing as sperm, if you leave the testis, is the epididymis. And it actually is the head of the epididymis, then the midpiece or the body, and then the tail of the epididymis, which is down below. And this again is all one tube. Remember, where did this come from? Something called the Wolffian duct, which was a long tube. The epididymis is a long tube. It's highly coiled. Okay? That means the tube has one end and another end. Where sperm enter is here on top, and then sperm move to the body, to the bottom, which is called the tail of the epididymis. The tail of the epididymis then turns into the vas deferens. The vas deferens is just a long tube that goes all the way inside the body, around the bladder, okay, and then fuses with the ureter from the urinary bladder, okay, from the bladder. Those two tubes come together, okay, and that fused tube, the urethra, then goes through the penis and out, okay. Now, just like maybe you learned about the GI tract, which is just one long tube. There were accessory glands, right, that put stuff in the GI tube, like the pancreas, for example, provides enzymes that go to the tube. There are accessory sex glands. Accessory sex glands that put stuff in this long tube are the seminal vesicles, the prostate gland, and the bulbourethral gland. So these are just three different gland structures that along the way make some stuff, buffer some nutrients, some minerals, and they put it all in the tube and it all gets into the ureter and eventually the urethra and is part of seminal plasma. Okay? Which of these structures are derived from the Wolffian duct? I'm helping you study right now. Lots of repetition. Epididymis, okay. Vas deferens, yep. What next? Seminal vesicles, anything else? Nope. So that's seminiferous tubules, and just like a garden hose is a tube, it has a lumen. Seminiferous tubules have a lumen. So if you think about where would sperm go? Would it go into the lumen or to the bloodstream? In lumen, thank you. Okay, not the bloodstream. Really, last thing you want is take a blood sample, you look on a microscope, you see a whole bunch of sperm. You've got a serious problem at that point. Okay. Testosterone, on the other hand, hormones, most of that hits the bloodstream. Now we'll talk about the fact that testosterone also will go to the lumen, but that means it needs to go 2 different places. Okay. Once it goes to the bloodstream, where does it go? Well, the rest of the body. Okay. Any questions about this? Makes sense, right? It's just anatomy. Okay, it's just words. Okay. Okay, testis function. We kind of talked about this, okay? Very early on, most boys recognize really quickly when they learn how to bike on a bike that has this stupid bar in between, that their gonads are not inside, but they're actually dangling in a scrotum, okay? And very early on, you start wondering why, oh why is it there, okay? There's a reason for this, okay? For the testis to function properly, it cannot function at 37 degrees Celsius, which is core body temperature. It only functions a couple of degrees lower. So how can you, if you have an organ that only functions properly at a lower temperature, what can you do? Well, get it out of the body, and that's exactly what happens. Okay, so the 2 ways that evolution has kind of dealt with this. This is 1, push it out of the body, testicular descent into a scrotum. And the second is something called pampiniform plexus. The pampiniform plexus is a countercurrent exchange of blood vessels. Okay? And this countercurrent exchange ensures that the blood that actually finally reaches the testis is already cooled down. So a picture of that is shown in this slide right here, which was same as the previous one. But it's this whole collection of testicular veins that starts wrapping around testicular arteries. So what happens here is that we have blood coming from the body that goes towards the testicular arteries, and that blood is 37 degrees Celsius. It's warm. The first thing before it hits the testis, what it sees is a whole capillary network of testicular veins that is cold. It's returning blood from the testis back to the body. So basically, the blood is already cooled down to some extent. Okay? So that's the first part. The second part is testicular descent. Okay? So testis descent is an important process. We just talked about this woman with undescended testes, and I highlighted the necessity to remove these undescended testes because they can become cancerous. And on top of that is it causes infertility. It destroys the germ cells and things like that. Okay, so testicular descent is important. It happens normally during fetal life, and normally when a baby boy is born, the testes are descended. Now, the one thing that has changed in the past 2 decades is that more and more the incidence of boys, baby boys that are born with undescended testes has increased quite dramatically, such that in some countries it is 1 in 10 baby boys that are being delivered that have undescended testes. Okay, so that is extremely, that's 10%. Okay, normally you would wait like maybe up till 9 months or a year. If it still hasn't descended on its own, you just go in and the doctors will just quickly pull it out of the inguinal canal into the scrotum and make sure that it stays there, okay? But it has to happen for the reasons that I mentioned before. Testicular cancer and infertility, okay? So how does this happen? Well, here's going back to the drawing, okay? There is this ligament called the gubernaculum, okay? So early on, before anything is descending, okay, but we form the testis and it does its thing. The testis and the ovary are the same spots. They are held in place by a ligament, a suspensory ligament, and a gubernaculum. The gubernaculum just attaches it to the body wall, okay? What happens is the testis in fetal life secretes hormones. We talked about that. Testosterone, right? AMH. There's a third hormone that the testis now secretes. about the testicular descent, this part? Yes?
So has the ISN always been secreted from the Leydig cells, and you just told us that now, or this only starts now?
Yeah, so it starts after the testis has formed, right? So then INSL3 will also be secreted. Okay? Yes?
Could you repeat with the transinguinal?
The transinguinal migration, all I want you to know there, that it's basically the gonad moving through that little tunnel in the body wall. Okay? Cool. Okay, let's quickly look at what a cross-section of a seminal vestibule looks like. Okay, again, think again of a garden hose that you cut through and you kind of look inside. Okay? So this is a cross-section of a seminal vestibule. Remember, we talked about the fact that at this point we have two types of cells in there, Sertoli cells and germ cells. Okay? And there's a lumen. Okay? It's a tube. What is this stuff here? Tails of what? Tails of what? Sperm. Yeah, so we have sperm that is being released into the lumen of that tube. Okay? And so basically, this is an epithelium. Okay? See all these little marbles that are there? Different sizes? Those are all different germ cells. So we're going to give them a different name in a minute. Okay? We're going to give male germ cells a bunch of different names, just like we give undergraduates different names depending on how mature they are. Right? Freshmen, sophomore, juniors, seniors. There you go. Yeah, I know there's a whole bunch of other names I could have come up with as well. I played it fairly safe. Okay? Germ cells are the same way. Okay? So this is a basement membrane. This is a lumen. All these, the immature, the freshmen, we like to put the freshmen in the basement, right? They're down below. No view, there is no environment, there's no sunlight, it's all good. Okay, so they're in the basement membrane. When they start maturing, okay, there's a rite of passage almost. You get to move a little closer and closer and closer towards the lumen. At some point, you finally graduate, you're done, and now you're sperm in the lumen. Okay? So germ cell maturation goes from immature here to there. Okay? In between all these marbles that are germ cells, we have Sertoli cells. And I appreciate, just like everybody else, that neurons are amazing looking cells, especially when you start labeling them with beautiful colors. Astonishing, really cool. I do like to make a case for Sertoli cells. Okay, so this is what one Sertoli cell looks like. This is the basement, and this is the apical side, or facing the lumen. So one of these cells, the basement is here, it stretches all the way towards the lumen. Okay? So the length or the thickness of this epithelium is dictated by the Sertoli cell. Okay? And so in between these Sertoli cells, we have all these germ cells. And that's where all these pockets are. Okay? Remember, Sertoli cells came from what we called somatic support cells. That support is really both physical, like all these little pockets, and by providing nutrients. Okay? Does that kind of make sense? Okay, this slide really doesn't add that much to it. It's just another picture. Here's the basement membrane. These are all these germ cells that are growing up and maturing. So this one gets a little older, turns there, older, older, and bloop, in the lumen. Okay? No Leydig cells here, but they're kind of in between the seminal vestibules. Okay, just a quick summary slide of what Sertoli cells are and do and things like that. And a lot of this we'll talk about on Monday, okay? The first thing is that the Sertoli cells form tight junctions. I know that Dr. Warren talked about gap junctions and tight junctions, adherent junctions, all kinds of junctions, okay? The tight junctions, think of it as the Sertoli cells that I just showed you, these awesome looking tall cells, holding hands, okay? They're all holding hands together, and basically by all holding hands together, they kind of form a barrier and two compartments within that epithelium, and we'll talk about that. That actually forms the blood-testis barrier, and that's an important barrier, okay? It supports germ cells, I mentioned that. It secretes fluids, okay? Obviously, the lumen is not a vacuum. There's fluids that are being secreted, things like that. It secretes a peptide hormone, inhibin, that we'll talk about on Monday. It has negative feedback to the brain. Any kind of crappy sperm, it tries to phagocytose and kind of recycle some material or get rid of it. It secretes not a hormone, but a protein called androgen binding protein. I love that name. It actually says what it does. It's a protein that binds androgens. Okay, and that gets released into the lumen. Okay, and then finally, in fetal life, it secretes AMH, and we talked about that. Okay, last slide for today. I still have 5 minutes, so this is an important one. I just told you, just like we give undergraduates different names depending on how mature they are, right? Freshman, sophomore, blah, blah, blah. We do the same with germ cells. So this part is terminology, okay? So keep picturing the seminiferous epithelium in front of you. And we start at the basement membrane, okay? The ones that are closest there, or the freshmen, are spermatogonia. That's the name for them. They're spermatogonia. How many chromosomes does a human spermatogonium have? 23? 46. 46. Okay, we start with 46. At that point, it's just like a somatic cell, 46 chromosomes. Okay? Now, at some point, the freshmen, they finally graduate and they become sophomores. In this case, the spermatogonium decides to enter meiosis. Remember from life science and high school and I don't know what else other class, when they talked about meiosis, there were always two. Meiosis 1, meiosis 2, right? First we do meiosis 1. So the one that decides to enter meiosis is called a primary spermatocyte. A primary spermatocyte is a little hint here. Primary, 1, it does meiosis 1. Okay? So it does meiosis 1, it finishes meiosis 1, and that is like graduating as a sophomore. Okay, so now you enter your junior year, and now you're a secondary spermatocyte. Guess what a secondary spermatocyte does? Meiosis 2, exactly, meiosis 2. You finish meiosis 2, and now you're a spermatid. Now you're a senior, finally. Okay, you're actually on the top floor, you have a view, there's sunlight, okay? How many chromosomes does a spermatid have? So now we've done 2 rounds of meiosis. Now we are haploid and 23 chromosomes. Very good, 23, okay? So, and then the only thing that has to happen at this point is a metamorphosis, so a change, where we're going to turn a round cell into something with a head, a midpiece, and a tail. Think of this, that transition, the spermatozoa. Those are your TAs, the graduate students. They're kind of hanging on, okay, for dear life. They're reluctant to let go, but at some point they will, and then they leave. It's like they're jumping off the building for various reasons, okay? Okay, so it's a quick summary. Spermatogenesis, the making of sperm, is lots of sperm by mitosis. Mitosis is the spermatogonia. Then making haploid, which is meiosis, and then the metamorphosis. Have a great weekend. See you guys on Monday. Yeah, always. What's up?
So I started this like beard growth protocol where
Yeah.
you put minoxidil on your face,
Okay.
which from what I understand like activates the DHT
Yep.
receptors
Yeah.
and causes masculinization.
Yeah, so it's basically it's a synthetic androgen that will activate androgen receptors. Yeah, yeah.
So